So I welcome you all for today's talk. We have Dr. Francesca, Cella, and she's a wonderful researcher. I've known her for quite a long time, and she's kind of here from the very early stages when this single molecule light sheets actually came up. So it's a wonderful an occasion to have her. I'll just talk a little bit about her. She is presently a assistant professor at the Department of Physics, University of Pisa, Italy. And her research mainly focuses on the design and application of optical microscopes, the techniques associated for mainly for understanding biological processes. In particular, her work expands on fluorescence microscopy, fluorescence imaging techniques, super resolution, computational approaches, light sheet microscopy. I think a lot on fundamental microscopy, and to understand cells, tissues, organelles, how they work out. A little bit of her background: she graduated in physics from University of Genoa and completed a PhD in physics from the same university under Professor Alberto Diasporo, who himself is a researcher in the field, and she has done a lot of research across the world. She worked at EMBL Heidelberg, which is European Molecular Biology Lab, which is really epicenter of microscopy, imaging, and fundamental aspects of biological sciences. She worked at also University of Maine and at University of Frankfurt, and it says a lot of research experience that she has. And so also she worked as a researcher at Italian Institute of Technology and Institute de Sciences Photonics in Spain. So a huge experience that comes in, and we are very happy to have her. We're going to learn a lot, because she herself has done a lot of single molecular microscopy as well as super resolution, light sheet. I mean, it's really. So, Fra, I will not go on and on. Can you please take over and please,
So, Partha, first, thank you for the nice introduction. And meanwhile, I'm trying to share my screen. Just give me a sec. Can you see the presentation right now?
Right, yes.
Okay, so sorry for the problem. So here, thank you very much. I was saying, Partha, for the nice introduction and for the invitation. So today we will, as you mentioned, we will talk about single molecule localization microscopy and the advances in the field and some possible applications. So the basic idea is that what we want to do is to actually observe biological processes. So my research interest was starting from the passion of trying to understand what happens inside. What happens inside the cell. So this is the animation from Harvard University. You probably already saw it, but it is a nice example of how much and how the cell is complex, the cell behavior is complex. So within a cell, we have to think about the fact that there's a lot of processes going on, building, destroying, molecules interacting.
Excuse me, Francesca.
See?
Sorry, can you please make full screen? Presentation mode.
Partha, can you hear me?
Yeah, I can hear you. Can you make it full screen?
This is full screen for me, right?
But we see the window below. I mean, now it's full screen. Yeah.
Okay.
Yeah, now it's perfect.
Okay, cool. So the basic, my first interest was trying to understand all these processes, and these processes are happening like polymerization or binding and unbinding of molecules. This happens on the nanoscale level. So we have also to think about, we are imaging matter, but we are dealing with the active system. So a cell is basically a system that is not only observing matter, like passive units, but we are actually observing active matter. So it's something that is far from equilibrium, if we have to think about it. So we need tools that can observe both dynamics and processes. So the basic idea, the starting point is to observe the structure of proteins and molecules inside cells and try to link it with the function. So trying to match this relation between the structure of proteins and the function in biological systems. So this opens the way to a lot of application. So this application can be like understanding which are the mechanisms involved in some conditions, like disease or pathological condition. It can be like testing and working in the field of nanomedicine is testing new therapies or therapeutic solutions, or also in the nanotechnology field is like trying to characterize bio-inspired materials. To do this, is important that we can access some information of the samples. In particular, how the molecules and the proteins organize. So what does it mean? This means if we have a few molecules that are building complexes, understanding their stoichiometry, for example, or if we have proteins cooperating, characterizing their clustering, so how these proteins are clusterized, or maybe how these proteins are organized in supramolecular structure or filaments. So all these things can be accessed only with a joint cooperation from microscopy, optics and photonics, molecular biology and chemistry for the sample preparation side, and computational approaches. So if we work at the edge of these disciplines, we can actually access these informations. And let's think about that in the panorama of the microscopy techniques. We are interested to image something that is below the diffraction limit, because the cells and the features in the cell that we want to image are in the scale of the tens of nanometers, like 20, 30, but this is the range that we want to image. So if we think about within the dimension scale we have in organisms and up to the molecule level, we are actually interested in this range that goes from few nanometers to hundreds of nanometers. And if we think about the microscopy techniques we have available in up to date, we have different kind of different tomography, resonance, everything. But optical microscopy is the one that touches this level. And basically, we can go and have lower super resolutions, or resolutions with electron microscopy. But in this case, we have the drawback that we are confined from the sample side. So we want something that is compatible with live cell imaging also, that is specific, so that we can label a specific protein and follow the fate of this protein. And we need super resolution. So the only one technique that matches all these informations is super resolution microscopy. And this is because it provides not only the resolution, but also the specificity. So we can label with specific fluorescent markers. And we can also work with live cell imaging, so we can follow molecular dynamics and interactions and not only structures. But now we will see also how we can improve this technique. So which are the other advantages we can exploit? For example, optimize it to extend it to three-dimensional samples and not only to the whole cell level. So for the specificity, we have different options. We have a broad span of fluorescent probes that we can use, but we always have to think about that we are actually working with optics. So we can combine all the advantages and bring the advantages of optics microscopy, but also with the background where we have a broad range of fluorophores that we can use. But let's see how does it work, the techniques, because in choosing these fluorescent probes, we need to put some care and attention, because not all probes are suitable. But let's see how does it work. I will tell you briefly the basics of the single molecule localization techniques, so we can understand which are the requirements. So the basic idea is to circumvent the diffraction barrier and the Abbe's law. So usually you cannot resolve objects that are closer than a certain limit that is given by the Abbe's distance. But if we play somehow a game, we can, this is taken from the literature, this is one of the first images published in the literature. These are microtubules, wide field imaging of microtubules, and this is the effect you can gain with super resolution. So how you can reach this result? The basic idea is that we are actually imaging our sample that contains features, sub-resolved features that we don't actually know a priori. We are imaging with the system, so we have finite point spread function given by the microscope we are actually using. And if we image all the molecules together, we cannot appreciate any detail below the diffraction limit. But this is different if we manage to play with the photophysics of the molecule and have only sparse subset of molecule on in our field of view. In this case, what we can do is to localize with different approaches, different procedures. This, from the computational point, we can discuss which is the best way to localize, but we can localize molecules and put the center and identify the center of each molecule with nanometric precision. Then we can actually play with the fluorophore and the lasers to turn them off and turn on other molecules. So if we play this game multiple times and we continue localizing the molecules, we actually end up with a super resolution map. So we end up with an information that is diffraction limited and below the diffraction barrier. And we did not actually change the point spread function of the system, but we finally have the information we wanted. And this brings an information that is definitely super resolved compared to observing the whole molecules together in time. But I was saying to you that we have a lot of fluorescent probes. Actually, we have different fluorescent markers, and we have to consider that they have a size of few nanometers, but we are using a common microscope. So what we need to do is to switch molecule on and off. We have to identify two states, an off state and an on state, and we need fluorophores that goes back and forth from these two states in a controlled way. And this can be done only exploiting the characteristics of the fluorophores in terms of photophysics. So basically, these are the transitions that the fluorophore can have. And the molecules goes from the ground state to the first excited state, and they emit photons due to the fluorescence process. So in this cycle, actually, we can, for example, this is the simpler way, it's not the only one, there's other, but this is one, and one of the simplest, is to bring molecules to some dark states. For example, the first triplet excited state. This is a dark state where if we push the laser to higher levels, the molecules at some point, even if this is a forbidden transition, the probability is not non-zero. So at some point, molecules will go to the triplet state. And the residence time in the triplet state is longer than the one in the singlets. So at this point, we have the molecules parked in a dark state, and few of them are going from the ground state to the first singlet state to emit fluorescence. So then they goes to the triplet and they go back from the triplet and the cycle is repeated. In this way, we can reach the sparse regime we were mentioning. But to do this, let's think about that we need to play with the photophysics, but we need also to play with sample preparation. Because not all the fluorophores can do this cycle, go to the triplet, stay in the triplet for a longer time. There's some limitations, but there's something that we can do to help this process from the chemical point of view, that is changing the environment. So using buffers that allow these transitions or favorite this transition. For example, we can use oxygen scavenging system, because if we have an oxygen scavenging system, molecules goes to the triplet, but they don't bleach, because in the triplet they are in an external shell and they are more prone to bleaching. So if we play with the environment, we can tune the perfect situation and condition for the fluorophores we choose. So let's think about when we plan the experiment, choosing the fluorophore then means creating the environment for this fluorophore to blink, so to make single molecule imaging. These are just an example. These are microtubules labeled with Alexa 647, blinking, and this is when we reconstruct the image, the result. As you can see, it's color coded, so it goes from green to red, because it's also like a 3D technique that with, for example, in this case was done with, we will see applications done with astigmatisms. This is the resolution, the result of localizing also in the axial direction using the astigmatism procedure. This is basically done inserting a cylindrical lens that creates distortion. So you have an elliptical spot, your point spread function is elliptical depending upon the axial position. So if we decode this information, we can map the axial position. Choosing the right, planning the right experiment is also thinking about choosing fluorophores that can provide the best localization precision, because what we want to do is to be precise enough, and this depends on how many photons we are collecting. So let's try to maximize the number of photons, both with the buffer and with the fluorophore we choose. And we need to reduce the background, because the background noise will ruin our localization performance. So in this case, this also, all these rules are actually valid also for the live cell counterpart of single molecule localization, because when we do single molecule localization, it can also be in live cells, but basically we are focusing on structures. But most of the time, if we have some dynamics or kinetics, actually we need to follow and do some single molecule tracking more than single molecule localization, and this is the counterpart. This is an application we were like studying RNA polymerase within bacteria in E. coli, and actually we were able to track the RNA polymerase inside, and we could like observe different tracks conditions, velocities, and actually sites within the bacteria that were accessible by changing the medium and treating the bacteria in different ways. So this brings the fact that RNA polymerase is confined in different compartments and move within different compartments. So now let's see how we can optimize this technique for addressing biological questions. I was mentioning you that one point is moving to 3D imaging. So this technique is well established at the cellular level, but now we will see how we could go to thicker samples. So basically, We can play to move from single cell level to tissues, for example. So we are dealing with scattering sample at this point, like brain slices, retinas, or we need to think about 2 points. One is how we are handling our sample, so the procedure to hold the sample. And on the other side, we need to think about, this is a scattering sample, we need to reduce the light sample interaction to reduce the scattering effects. To improve the imaging, the single molecule imaging in tissues, we can play on different sides. So one is the optical approach, so changing the optical approaches we are using, the architectures. One is working on the fluorophore and the sample preparation side, using optimized fluorophores. And the other one is the computational side, where we can help with new localization algorithms, for example, with stronger performances to be more robust in tissues. So first, playing from the optical architecture side, we can think about combining single molecule localization with the 3D techniques to extend the imaging depth capabilities. The one actually we choose was light sheet fluorescence microscopy. So you probably already know this technique, but I will give you some basics. The basic idea is that we are not using, like in the single lens based methods, the same optics. We don't have the same axis for illumination and detection, but we are decoupling the illumination and the detection. They are oriented at 90 degrees. This means that we are creating a planar illumination, so we are creating a plane of light, and we are actually acquiring the signal at 90 degrees. So we are doing an intrinsic optical sectioning in this way, imaging this plane within the sample. The other advantage is that this is a fast technique, because it can be a CCD, a camera-based technique. So it has also the requirements that are in common with the single molecule localization, that is fast imaging speeds. Then we have an intrinsic high signal to noise ratio, because we are actually confining the illumination. So we are removing the contribution above and below the focal plane. So the out of focus contributions are reduced. And this means we are reducing the background. That is something we want for single molecule detection. And we have a low photo damage. Actually, this technique was actually discovered in the early 30s for different applications, but it has been widely used in the last decade in the developmental biology field. This is just an example. This was acquired, this is a paper taken from the literature, but this was acquired when I was at the EMBO. So a colleague of mine was acquiring this amazing video. So I was like impressed, and I fall in love with this technique when I saw this video. Here you can see the development of an embryo. So from the first cell stage level, they injected H2B GFP, and then they were observing over time how the embryo is developing. So I was really impressed by this technique. And the idea was, okay, why I don't combine this method with single molecule localization to bring benefits to the single molecule localization technique. So the basic idea was building a system. Here you can see the single plane illumination microscopy, point spread function. So basically we have the light sheet profile, and then we have a detection that is oriented at 90 degrees. So the overall point spread function of the system in the end is more isotropic compared to a single lens based system. So this brings the reduction of the background actually. So for single molecule localization, this is also a huge advantage, because if you think about when we work with wide field, we are actually exploiting all the molecules in the focal volume, and we are turning on and off the molecules in the whole volume. But if we use the light sheet microscopy, what we can do is to confine the illumination, and we prevent the molecules above and below the focal plane from switching, so turning on and off. This means these molecules will be later on available again for another image. So if we move the plane, we can perform a single molecule localization stack. So this was the first system we built. This is a conventional selective plane illumination microscopy. We have visible lasers that can be combined, but also 2-photon excitation laser. So the basic idea was having a system that could work both in the visible but also in the infrared range to bring additional improvements. And then we can combine all the illuminations within a cylindrical lens that focuses the back focal plane of the illumination objective that creates the light sheet within the sample. The detection is basically a wide field detection scheme with some additional magnifications for single molecule detection that are needed for proper single molecule detection and localization. So here you can see the first sample I was approaching. These are mammary cell spheroids. These are objects that are, we moved from cells that are 10 microns, let's say, to objects that are hundreds of microns. So these are cell aggregates that are definitely one scale bigger. But since due to the size of the sample, we actually have to think about which are the effects induced by scattering, both along the illumination path and also along the detection path, because light travels twice. So this is the image. Here we were actually expressing H2B cherry, that is a fruct-activatable fluorescent protein suitable for single molecule localization. And we could localize histone within the nuclei, and we could reach a theoretical localization precision of 28 nanometers. So basically, just saying a few words about the localization precision, because this was a huge part of the characterization of this work, because when we talk about localization precision, and I'm saying, we reached 28 nanometers, I'm talking about theoretical localization precision, because this is a localization precision that has been estimated with an analytical model. Here you can see the models I was using that is commonly used, that is the Mortensen's model, that I was mentioning you before. It contains some experimental parameters like the number of photons, or S, that is the point spread function, the standard deviation of the point spread function of the system, the pixelization parameter, that is the provided by the effective pixel size of your system, the background. So all the experimental parameters are considered within this analytical model, but we have to think about this is only a model. So this model has been developed and it works fine for transparent samples. But when we move to tissues and light sample interaction are stronger, we need to consider some more factors. So we need to consider that we have some real localization performances that can be quite different from the theoretical ones. So in this case, we were actually considering a model in which we were considering the effective localization precision. So the effective localization precision was given by this relation that contains a term that is the theoretical localization precision, that is this sigma localization, plus another term that is called sigma instabilities. So this factor contains all the instabilities and defects that are given by the light sample interaction that can ruin our theoretical localization precision. And then we went to measure. So we went to inside the mammary cell spheroid. We found some sub-resolved and some molecules, and we were doing repeated localization of these molecules, building an histogram of the positions along the radial and along the axial direction. And then we estimated the localization precision as the full width half maximum of this experimental distribution. So we saw that the theoretical values that were 26 and 60 in the axial direction, in the radial and axial direction respectively, if we measure directly within the mammary cell spheroid, so we are hundreds of microns inside the sample, these are actually goes around values that are twice the theoretical ones. So we got 63 nanometers as the localization precision along the radial direction and 141 along the axial direction. What does it mean? Which is the take-home message here? We still have super resolution. This is still a good value because we are imaging tissues, so it's fine. But let's be careful because considering the theoretical localization precision is probably simplistic and does not give a real picture of the real situation. So let's measure and consider that our localization precision and the resolution as a consequence are higher than the one that we could estimate with the common models. So this is mainly due to scattering effects, and scattering effects can occur along the illumination and along the detection path. So first, let's start to characterize this effect. So let's see what happens in the detection path. So if we have a mammary cell spheroid, we will image deeper and deeper into the spheroid. And actually we will go into the spheroid checking that the localization precision is not cell dependent or depth dependent. So we perform measurements at different heights and we checked that the localization precision is depth independent. And this is the case. If we look at the histograms, the number of photons we could get is more or less in the same range, even if we are going deeper. So from this side is fine. The other side is checking that we don't have aberration effects and checking, this is another check that we have done, that the light sheet does not lose intensity traveling into the sample. So we checked all these factors, and this is the result. Here we are imaging connex in 43 within the mammary cell spheroid, and we choose a cell, we focus into a cell, we perform plane by plane super resolution imaging. These are displayed in a space 1 micron, one from each other. And then here you can see a color-coded map that make the summary of all the contribution in the different planes. But these are super resolution contributions, so we could do a super resolution 3D stack. So another option, another point I was actually optimizing is how to use this method to be a quantitative tool, because we are accessing information at the molecular level. So what we want to do is to be quantitative, and we want to exploit this technique not only to be an imaging tool, but also a quantitative tool. So we have different options. One is molecular counting through stepwise photobleaching, for example. And the other one is performing single molecule localization microscopy and clustering analysis. So if we focus on these two ways, the first one is okay, it's fine, but it works if you have few molecules. So up to 5, 6, 7 molecules, you can try to count the molecules by the photobleaching steps. But if this number, this works for receptors, for example, glycine receptor. But if we want to go to other quantitative tools, what we need to do is to perform clustering analysis. And then we need to convert the histogram we can get, this is the histogram of the number of localization per clusters, into the number of protein of interest. But this has a limitation because we need to think about that we have labeling and counting errors. For example, we need to consider that there's overexpression, blinking that can go or provide overcounting. or we can have incomplete protein maturation if we are using protein, or uncontrollable stoichiometry if you are using antibodies, that can make overcounting or undercounting. So this means that we need calibration standards. So for example, we use DNA origami as a calibration tool, so we could attach the number of proteins we want, and we could characterize the histogram of the number of localizations in this case. This provides us a strong quantitative tool, because we can convert the number of localization into the number of real proteins or protein of interest. But let's see which are the possible applications. I'm just giving you a few ideas here, a few examples. One is, for example, studying molecular motors involved in the intracellular trafficking and transport. So the basic question was how many motors, how many dynein. Dynein is a motor that is involved in the intracellular transport. It works on microtubules bringing cargoes. So the basic idea is that how many motors are required to bring a cargo. So first we label the cell, we label dynein within a cell, and we perform clustering analysis. And if we look at the result, here is the portion of the cell, you could see regions where you see small clusters, and it seems like a sparse clusters. But then there are regions where you could feel like there's something different going on. So we decided to sort the dynein, dividing it into categories: engaged dynein and dispersed dynein. Engaged dynein is the dynein, we selected the dynein on top of microtubules. So these are the dynein that are supposed to work on microtubules and eventually bring in cargos. We don't know, but at least we could know that these are candidates, functional dynein. The other one are dynein that are within the cytoplasm, doing probably nothing or waiting to be recruited somewhere to do something. So this sorting on dynein on microtubules and dynein not overlaid to microtubules provided different distributions. So we perform clustering analysis. You can see from the clusters here that it seems that there's a different structure and bigger cluster on top of microtubules. So we performed the clustering analysis. We could get the histogram of the number of localizations. And if we look at the number of dyneins, then you can see that we have different numbers, and we have only one dynein in the cytoplasmic case. So the dispersed dynein is single dyneins, but the one overlaid on microtubules are more dyneins, and within a range that goes from 1 to 7. So if we look more in details, and here you can see clustering analysis and the difference. Here you can see the structure for the engaged dynein, the structure of the microtubules with the clusters on top. What you can see here, that the distribution of engaged dynein, so the dynein that probably is bringing cargos and works on microtubules, is actually a distribution with a peak on 2 motors, while the dispersed and cytoplasmic dynein still remain 1 motor. And this is a quantitative information. We are actually calibrating with DNA origami and counting dyneins. So this is not the number of localization on the or number of fluorophores. This is the number of motors we are actually counting within the cell. And this is the information we wanted. Another option is trying to understand which is the role of cellular adhesion when cells lays on different substrates. So here you see neurons, and these neurons are laying on glass and graphene. And in the literature, it has been observed that cells prefer graphene, and they work on graphene at some point, growing. So why the cells prefer graphene? So which is the role of the adhesion process? So we choose vinculin, that is a adhesion protein that stays in the first transduction layer of the adhesion complexes, so in the intermediate layer. And we could observe, we went to study the distribution of vinculin to understand the addition on glass and on graphene. So we performed in the first line, this is on glass, the second line is on graphene. We performed storm and the single molecule localization imaging, clustering. And already from these images, you could see a difference in the distribution. So the focal additions looks more compact on graphene than on glass. So if we try to quantify, we could quantify the number of localization per clusters and the cluster area. And here you can see differences from glass and graphene. We could see that there is an increasing, both on graphene, there is an increasing of both number of localizations and area and density. So this means that density of localizations within the clusters on graphene is higher than glass. So this happens in mammalian cells. So this is CHO cells. But what happens in neurons? So what happens in different cell with different functions? We repeated the same experiment and we went to estimate the same parameters. And surprisingly, in neurons, this was not happening, or well, it was slightly different from the mammalian cell case. There was not a direct increasing in the density and in the number of localization on graphene, but neurons still, if you plate neurons on glass and graphene in a patterned way, neurons prefer, actually they prefer graphene. So we went to understand further the nanoscale organization of vinculin, and we found that within each cluster, there's no improvement, increasing in the number of localizations. So does not mean that each cluster becomes more populated by vinculin, but we have a higher number of smaller clusters. So this means there's a fragmentation. So in graphene, from the overall vinculin is higher, but is organized in a different way. It is fragmented in smaller focal adhesion spots, but there are more focal adhesions and focal clusters in this case. So this means that from mammalian cells and neurons, there's a different nanoscale reorganization of adhesion proteins or vinculin that we were observing. But the nanoscale organization is quite different from neurons to mammalian cells. So just a few words about the fact that a last option is trying to understand what happens when we are doing different therapies, for example. So here you can see these are cells treated with different radiotherapeutic approaches. So we have usually radiotherapy is used in cancer treatment. Conventional radiotherapy has some drawbacks because at some points it destroys both the cancer tissue but also the healthy tissue. So for some like brain cancers or other cancers where you cannot destroy the healthy tissue around is not usable. And the flash radiotherapy is a new approach that can confine in time the pulses of irradiation, and it seems to preserve the healthy tissue more than conventional radiotherapy. So we tried to understand which is the damage induced by flash radiotherapy and conventional radiotherapy, and if there's a difference between conventional and flash. What we saw actually is that if we label phospho H2AX, so phosphorylated histones, we could see that there's this is a marker of DNA damage and early repair signaling. We could see that the number of clusters is actually similar. So here we were doing storm and clustering analysis. You can see overlaid on the wide field, there's the clustering. So the number of focal or focus or foci, I would say, is the same in the cell. So basically, it seems that the damage and the repair is the same because the number of double strand breaks, so the damage associated to each foci, the number is the same. But what happens within each single focus? So the number of phosphorylated histone within each focus is the same because in the cell, they're the same number of foci. But what happens at the molecular level? So we went to estimate the number within each cluster, the number of protein. And this is the clustering result, comparing conventional and flash radiotherapy. So what we can see here is that we have a different number of phosphorylated histone within each cluster and each focus. So even if it seems that the number of DNA double-strand breaks are the same in the cell, the response in terms of phosphorylation of histones is different. And this means that the damage and repair process are different in flash and conventional radiotherapy. So this is the starting point. This is early results from the experiments. What I want to give you as a take-home message is that we can use all this technique to move to tissues and to be quantitative. And this is something we can do that goes beyond having a super resolution imaging technique. So super resolution is nice, it provides nice images, but we should not stop at the images itself. We should try to, go further and try to use it as a quantitative tool or to push the capabilities going to, for example, going to imaging tissues in thinking about the future applications. So I would like to thank you for your attention, and I would like to thank for inviting me here, and also to thank all the people I'm collaborating with in the past and today. So thank you. I'm happy to take questions.
Thanks a lot, Francesca. It was a wonderful talk, and we could get a great insight into the type of work that is being pursued in your lab. Really enjoyed. Also, one thing that you can, as a participant, you can understand from a talk how to approach and address a problem using super resolution techniques or any other new technique that comes into existence. Okay, so that's how you approach a problem, and you can see so many plots and so many graphs. That essentially is how you quantify. The research is also a lot about quantification. Okay, and this is something real. This is how you go about solving a problem. Okay? So, again, thanks a lot. But I want to ask if you have any questions that you want to know from biological perspective or from physics, whatever.
I think there's one in the chat that I see right now. Partha.
Okay.
There was a question that is, which transfection method did you use for H2B cherry and spheroids, single molecule imaging?
Yes.
So while that was, this is a nice question because we actually had a bit of struggling on this side. We were doing transient transfection. And we both used, this was in collaboration with a cancer center in Milan. They were doing both lipofectamine transfection and injection. They were using the gun. They were testing different protocols. All of them were actually working. In the end, I think the measurements I showed you were the injection ones. But we have to think about the fact that this was a transient transfection anyway. So there was we had a window in which we could perform the imaging. So there was a time window in which expression was optimal. That was the drawback. Okay.
Okay, thank you, Frank. Okay, so do you people have anything that you want to ask? Okay, Francesca, I do have something that I want to ask you. What are the challenges do you see forward for deep tissue imaging as far as super resolution is concerned?
You mean as an application?
Yes, yes.
Oh, well, what I can see is probably since, bringing these techniques to the surgery is kind of difficult, okay? All the people are asking me, can I go to the patient in the surgery room? No. But what we can do is trying to make precision medicine, for example. So taking ex vivo or like histological parts and bring them to the lab. But this is something is feasible. The other option is trying to do non-invasive studies, like taking cells from the patient and growing cells in some bioavatar, like spheroids that can, where you can put also the cells from your patient and see the interaction of the cells of the patient with the other cells. Like, for example, if you want to test the immune system response, you can have your 3D spheroid, and then you can put the cells from the immune system of the patient and see how these infiltrate inside and how do they behave. So basically, the idea is that we cannot bring super resolution to the patient, but we can try to use super resolution with the cells or tissues derived from the paper and bringing them to the microscope and the lab, trying to do optimized observations. And this goes to towards precision medicine or personalized medicine, maybe.
Also, just wanted to know how deep we can go using, super resolution or super resolution along with light sheet. I mean, practically, how deep we can go.
So, well, the question is depends on the sample. This is the right answer. To give you some ideas or rough estimation, if you are looking to spheroids, and if you grow spheroids in some specific ways to help transparency, you can get, with no clearing, for example, hundreds of microns. So 200 microns, no more than 200, to my experience. If you have brain slices, the scattering is higher, so the density is higher, and so you can. I never made to reach something more than 60 microns in brain slices, for example. So it's really dependent on the tissue you are imaging. If you do clearing, and it depends also if you treat the sample, because if you have a slice and then you treat it with clearing, then things are, or expansion microscopy, something that makes optical properties and scattering and absorption lower, then you can go deeper, definitely. But you need to work on the sample side to make it more transparent.
Yeah, so, if anyone has anything else to ask, you can still go ahead. Otherwise, so I don't see anything, Tra. I mean, it was really a great talk, and we could gain a lot from it. And thanks a lot for delivering the talk.
Thank you very much.
Yes.